Okay, I think we should start. It is my pleasure to introduce Dr. Graham Pitcher, a distinguished cellular and systems electrophysiologist at Sick Hospital in Toronto. Dr. Pitcher's research is dedicated to understanding how information is transferred and processed at glutamatergic synapses in the central nervous system, with particular focus on NMDA type glutamate receptors. So his motivation stems from two key objectives. One is unraveling the neurological basis of human behavior and identifying therapeutic strategies to prevent or treat central nervous system disorders, which is the second one. Currently, Dr. Pitcher is investigating pain processing in the spinal cord dorsal horn, as well as mechanisms underlying learning memory and cognitive function in critical brain regions such as the hippocampus. His impactful research has been published in leading peer-reviewed journals, including Pain Medicine, Frontiers in Synaptic Neuroscience, Neuron, Cell, and Nature Medicine. Please join me in welcoming Dr. Graham Pitcher.
Thank you so much. Thank you, Dr. Faxmwali. I really appreciate the invitation. I'm honored. Merci beaucoup pour l'invitation d'être ici avec vous. I'm very happy to be with you guys. It's a pleasure. And also, of course, thank you to the Department of Surgical and Interventional Sciences. I'm a researcher at Sick Kids Hospital, a research associate. And by training, I've studied behavior, pain and cognitive type behaviors, as well as electrophysiology. One of my main interests is in how neurons communicate. I've done a considerable amount of research in the brain, looking at the hippocampus, learning and memory, also in models of schizophrenia. Presently, I'm looking at models in the hippocampus and the prefrontal cortex in depression. And I've also done research recordings, electrophysiological recordings in the cerebellum. So as you can see, I'm quite interested in how neurons communicate in health and in disease. One of my other major interests is in the spinal cord, and that is depicted right here. And as you can see here in this cross section, we have the dorsal horn where sensory inputs come in from the periphery, and also the ventral horn where we have motor outputs. My main interest is the synaptic connection between primary sensory afferents and neurons in the dorsal horn, specifically lamina 1. And my focus is on this connection, this glutamatergic synapse. And as we can see here, upon glutamate release, it activates primarily these two ionotropic receptors, the alpha amino, 3-hydroxy 5-methyl 4-isoxazole propionic acid, as well as the NMDA receptor and methyl D-aspartic acid receptor. I'll go into in a few moments a little more detail and how these function. Now, my emphasis, as I just shared with you, is the superficial laminate, in particular lamina 1, and that's primarily where we have nociceptors, pain receptors, if you will, that enter through the dorsal root into the superficial portion of the dorsal horn, form synaptic contacts, as depicted here. And this contact, these synapses activate projection neurons, which occur in lamina 1. And of course, these project to the brain and provide us with the perception of pain. And again, that's one of my main interests. Now, we have to consider the spinal cord with the amount of sensory innervation. It pretty much takes sensory input from the entire body. Now, why this is important to understand these basic mechanisms is because upon traumatic injury or disease, for that matter, one can experience chronic pain. And I'll just touch briefly on this. This I managed to get, it's from November 2024, just a few months ago, from the National Center of Health Statistics. And what they report on from their surveys in 2023 is that chronic pain, as you can see here, is quite prevalent. Here we have depicted females, males, and then the mean here. It's approximately 25%. And that's quite something. I've given a fair number of courses here at Sick Kids to nurses, medical students, and so on about basic pain mechanisms, chronic pain. And I was checking some of the slides I have from over the years, and it's actually increasing. Once upon a time, it was 1 out of 5 or so, to approximately 20%. But as we can see, the number of people suffering from chronic pain is increasing. And in fact, this added survey provided here, high impact chronic pain, these are from individuals who report having pain most days or every day. And this is quite devastating for them, obviously. So again, this is an impetus for us to pain research to make efforts to better understand chronic pain and come up with ways to treat it. And of course, come up with non-opioidal approaches to treating pain. So again, in that context, that's why I'm very much interested in the basic mechanisms of pain at the level of the spinal cord. Now, this little schematic, some of you may recognize it, is from René Descartes, 16th century philosopher, and he had certain perspectives on pain. And one of his views is that pain perception, as perceived here by a little boy with a fire, that this is transmitted or relayed to the brain through a fiber, a tube, a string. And at the time, it wasn't too far off. Now, obviously now, we've come a long way. If we consider McGill, Ronald Melzack the late Ronald Melzack made important discoveries in the pain center that exists there now, phenomenal top shelf work. But I would say in science, in the field of neuroscience, there is this view that pain perception does still function as a basic relay. That's what the spinal cord serves as, a basic relay. And what I'd like to share with you today is some of my recent work where I will propose that the spinal cord dorsal horn is not a simple relay system. In fact, it's quite a complex coding hub, which involves diversity in the glutamatergic synapse located in lamina 1, between primary afferents and the lamina 1 dorsal horn neurons. And this complexity, diversity, derives primarily from the NMDA receptor itself. So I'll speak a few minutes. I'll give you a basic crash course on voltage clamp recordings. The first part of my talk will be dealing with this. I won't go into too much complex detail, but basically this is actually a recording I made from transverse slices from adult rodents. This is one experiment. And we can see the inward and outward currents of AMPA and NMDA receptors here. And this is basically summarized on this current voltage relationship. I don't want to go into too much detail on that, but what I would like to share with you for a few minutes is this quadrant of the IV curve right here. This is potential here, negative going to positive, and negative current here, inward current. So if we consider our primary afferent, our lamina 1 neuron here, this is the synapse connecting them. Resting membrane potential is around -65, -70. And so when we have a presynaptic stimulus, let's say a nociceptive stimulus, that induces glutamate release, that binds to AMPA and NMDA receptors in the postsynaptic membrane. Now, given that this ion, sodium, is positive, the resting membrane potential is negative, and there's an abundance of sodium outside, you have an electrochemical gradient and a massive influx of sodium. Now, what's fascinating about the NMDA receptor, it's an aspect of it that we still study today, is that at these negative potentials down here, the NMDA receptor is blocked by magnesium. It's essentially plugged up. And how it becomes unplugged is with the depolarization induced by sodium influx from the AMPA receptor. So as the sodium enters, it induces a depolarization. It makes the inside of the neuron less negative. And so as we become less negative, you can see from this current voltage relationship with the NMDA receptor, an unplugging occurs. There's more NMDA receptor current. And as we become more depolarized, it unplugs. And at that point, calcium can flow down its electrochemical gradient and cause more depolarization. Now, as biologists, we all know that calcium is important for signaling. And with the amount of depolarization occurring by the sodium and calcium influx, that can activate the postsynaptic neuron and lead to transmission from the spinal dorsal horn to the brain where we perceive pain. Now, if it was that simple, that would be the end of my talk right now. What I'd like to share with you is that it is not that simple. There is a complexity, as I just mentioned, at individual glutamatergic synapses connecting primary afferents and lamina 1 neurons. And this complexity at individual synapses derives from complexity in the NMDA receptor itself. So how do I measure individual synapses? Well, I will share with you the schematic. If we consider this as a primary afferent in the dorsal rootlet, this is our lamina 1 neuron, and I'm recording it. I have my pipette right here, and I have a stimulator placed in the rootlets. And basically what I use is a technique as a minimal stimulation. And I think I'm probably one of the first to use it in the spinal dorsal horn. A few others, historically, if you look at work from Chuck Stevens, it was used in the hippocampus in the 80s and 90s, early 2000s, but very few, if any, have carried this out in the spinal dorsal horn. It's an incredibly challenging technique, but it is also quite exciting. And what I do is increase the stimulus intensity, depicted here, until I activate in the rootlet, in the nerve, a single axon. And when this occurs, this is the synapse here amplified. When I have a single axon activated, when I stimulate at that threshold, an action potential is induced, propagates to the nerve terminal, and activates calcium channels, which allow calcium influx shown here. And now, for those of you who follow presynaptic physiology, there's a complex array of calcium-sensitive proteins that lead to vesicular release of glutamate. And that activates, in this case, AMPA and NMDA receptors located postsynaptically. What's intriguing is that with this calcium influx, the release probability is actually very, very low. And it's not just the spinal cord dorsal horn. This is established at glutamatergics in many regions of the brain, such as the hippocampus. So with every action potential, you may or may not have glutamate release. It's a probability. And so I'll just show you an example, a set of experiments. This is from a recording from a single synapse, and to show what the evoked responses from minimal stimulation intensity, what these responses look like. And from the IV curve, as you recall, I'm holding positive. This is the AMPA and this is the NMDA component, and I'm holding at a positive membrane potential because this is without the magnesium blocking. You can very clearly see the NMDA component. So that's the purpose why I'm holding positive. So once I reach the threshold of activating a single axon, we see a success right here, a beautiful EPSC. And of course, for those of you who, well, if you're not familiar with electrophysiology, 20 picon, that's a very small response. That's consistent with a single synapse being activated. Now, as I mentioned, it's a probability of release. So in some cases, you give your stimulus, you get a failure. The calcium presynaptically at the active zone is not sufficient to lead to glutamate release. No glutamate release, no postsynaptic response. Simple enough. Another stimulus, you get release. So over a time course, this is approximately half an hour. This is what can be seen right here. These are this tight cluster here. These are the successes. And here on the bottom line, these are the failures. Increase the stimulus intensity just till I get to the threshold. Success, failure, success, failure. And incredibly difficult experiments to carry out. But my goodness, when I'm recording this, it's actually quite exciting. It's really, really fascinating. And this panel simply shows an overlay of a number of successes. And you can see that the amplitude is fairly consistent. And also the latency shown right here, consistent with a single synapse being activated, a monosynaptic connection, if you will. Here we see the distribution. This is a singular distribution. And I emphasize this point because if my stimulus intensity was a bit high and I was acting, say, for example, two synapses, two axons, two synapses, I would see another cluster of successes up here. And so we would have a double distribution. So again, this is very important to show that it's a single distribution, a single synapse unitary event. This panel shows here the EPSCs held at positive, the mean of these, and also at negative, because I can clamp at both positive and negative holding potentials. And what I can do with this mean is measure a variety of components of the micro EPSC, the half width, the rise time, the amplitude, the area under the curve, the decay, which is the time course of the decrease in current following agonist, in this case, a glutamate removal from the NMDA receptor channel complex. So how I got fascinated in this specific project I was carrying out, I was conducting these minimal stimulation experiments and carried out between 200 and 300 experiments. Again, challenging, but quite exciting. But what I discovered, which was quite perplexing, is that if we look at just a sampling of some of my recordings here, some of these recordings, you can see that they're quite diverse. Some of them have very fast decay kinetics. Some of them have intermediate. Some of them are quite long. And when I first saw this, I thought, man, there's something wrong with the amplifier, my recording. What is up with this? So quite perplexing. So I decided to carry out an analysis to see if I can figure out if there's anything I can glean, any information, if there's any classification from all of these. So I looked at peak amplitude. I looked at the rise time of each of these events, and recall that I have close to 300 of these events. For each experiment, as I showed you just moments ago, I had a measure of the failure rate, the input resistance. So in each recording, I could administer a pulse, and the input resistance that I was able to achieve from that gave me an indication of the health of the membrane of the neuron, essentially its health. I measured the decay, the half width, the distance, the time base between the rise and the decay, and also the charge transfer, which is essentially in voltage clamp area under the curve. I had all this information, but realized, how can I categorize it? I just cannot arbitrarily take these two examples, for example, and say, well, they're the same, I'll group them together. And this is when I sought the help of Dr. Livia Garcia, and a cancer geneticist, but with also expertise in computational biology. And we had already carried out some collaborations together. And so I approached her and I said, look, Livia, I have these very interesting responses, all of these parameters that I'm able to glean from these measurements, from the micro EPSCs, but I don't know how to classify them. And she said, okay, let's take a look. And so what she found, she carried out principal component analysis as well as hierarchical cluster analysis shown here. And she found that the charge transfer, area under the curve, the half width, as well as the decay is what makes these responses distinct, is how we come up with these, in her findings, 3 distinct classes of these micro EPSCs. All these other measurements, peak amplitude, rise time, they were all the same between the recordings. Now let's keep in mind the charge transfer, area under the curve, the half width, they're basically derivatives of the decay. The properties of the receptor that is mediating this decay, that dictates the speed at which it goes back to baseline. So that is actually the main in my opinion, what I discovered to be the main factor in distinguishing the EPSCs in these three groups. Now, with all these, with these three groups resolved, I calculated the mean of the decay in each one, and for the intermediate, we came up with a value of approximately 315 milliseconds at the time of the decay. For the fast decay, so you can see I termed it intermediate. The fast decay here, very fast, approximately 52 milliseconds. And with the slow decay, it was well over a second, which is quite phenomenal. Approximately 1,200 milliseconds, the decay. In select experiments where I had fast, intermediate, and slow decay kinetics, I applied the potent NMDA receptor antagonist. It's a competitive antagonist for glutamate on the NMDA receptor channel complex. And you can see here the fast decay completely wiped it out, left solely with the AMPA receptor response. Same with the intermediate decay and also with the slow. So what this told me is that we have three distinct glutamatergic synapses in spinal lamina 1 with distinct kinetics, but they are mediated by NMDA receptors. The question is, how can this be? Why is there such diversity in spinal lamina 1, and what exactly is mediating that? So that's when I considered in the field looking at work from Silvano Vicini, and he's a prominent investigator of NMDA receptors in the central nervous system and has done a lot of classification. And so in one of his schematics here, we can see that activating glutamate receptors can activate either metabotropic or the ionotropic fast-acting. And in this classification, we have the kinase, the AMPA, and the NMDA. And in the NMDA classification, we have a further breakdown of a variety of subunits. We have the GLUN1, GLUN2, which is further broken down into 2A, 2B, 2C, and 2D, and also GLUN3A and B. This is more of a recent discovery. So again, offering a lot of excitement to the field. What Pier Paoletti has done, another major prominent NMDA receptor physiologist, looked at all of these subtypes, predominantly the GLUN2A, B, C, and D, and most of the focus in the field, in fact, and not just Pier Paoletti, but many have looked at the role of these receptor subtypes in development from birth, juvenile, to the adult age in rodent brains. And of course, as you can see, there's very little on the spinal cord. It tends to be more focused on the brain. But what Silvano Vicini did some years ago, he asked, what role do these subtypes play in development? And so he carried out an interesting sequence of experiments where he transfected NMDA receptor cDNAs into HEK cells, human embryonic kidney cells, and recorded from them. Of course, this is a single cell. It's not a synapse. So to mimic the synapse, he applied glutamate. And in HEK cells where he had 2A, he had very fast kinetics, around 50 milliseconds. In the 2B, around 300. And in the 2D, approximately 1.7 seconds decay times. Now, if you recall from my recordings just a few slides back, it's actually quite comparable. 52, 315, I think I recorded in around 1.2 to 1.3 seconds. So what I considered at that point is that, well, could it be that in my experiments, the differences in the responses that I see between the three synapse types, could it be, we know it's NMDA, DAPV blocks each one. Could it be that the part of the diversity we see here is due to GLUN2 subunits? So with that in mind, I carried out another sequence of experiments where I measured fast, intermediate, and slow kinetics and administered selective allosteric antagonists of these receptors. And so I began with a DQP1105, which is an established GLU-N2 inhibitor. And so on responses, micro-EPSCs with fast kinetics, it had no effect at all, shown here in the scatter plot, baseline versus administration of the drug. In micro-EPSCs with intermediate kinetics, displayed here, shown here and here, see absolutely no effect. However, in micro-EPSCs with the super long, prolonged kinetics, you can see a dramatic effect quite clearly here in the trace and also in the summary. And these histograms basically show the same data just with statistics. So what this tells us in this first set of experiments is that the micro-EPSCs with the slow, long kinetics are probably mediated by GLU-N2D. It's the same similar approach, set of experiments, but in this case, I used row 2569881, which is a known GLU-N2B antagonist. And you can see with the fast decay micro-EPSCs, absolutely no effect. With the intermediate, a dramatic effect right here. And we also saw a partial effect on the slow micro-EPSCs. Suggesting or indicating that the GLU-N2B NMDA receptor subunit mediates primarily the intermediate micro-EPSC. And of course, the fast micro-EPSCs, which were insensitive to the Rho compound 2B and the DQ compound, the GLU-N2D, was exclusively sensitive to the GLU-N2A antagonist TCN201. You can see that right here. So what that tells us is that these three groups that we discovered, the heterogeneity, the complexity is mediated primarily by the NMDA receptor GLU-N2 subtype present at the synapse. So in this case, the fast is mediated by predominantly GLU-N2A, the intermediate by 2B, and the super slow by GLU-N2D. And I'll take a few minutes just to describe to you how exciting this was upon this discovery, because as I said, I've done recordings in the prefrontal cortex, a bit in the cerebellum, quite a bit in the hippocampus. And as important as these brain regions are for learning, memory, complex mental processes, in my recordings, I've never seen in a given synaptic population or pathway such complexity. So right off, I hope you realize that the spinal cord dorsal horn in a region, the superficial laminae, it's quite a complex region. The synaptic transmission is quite complex, more complex than I would say previously thought. So in voltage clamp, the recordings I just shared with you and depicted here, you're holding out a voltage and recording the current. So that gives you a measurement of the current, and from that you can learn a lot about the properties of the receptor synapse in question, and as I just shared with you with the different subunits of the NMDA receptor. But it doesn't tell us very much about function. One of the unique abilities in these recordings, at least the recordings I do, with the recording pipette specialized intracellular solution, I'm able to convert my recordings from voltage clamp to current clamp. And this is more of a realistic measurement, because it's here you're clamping at a voltage, looking at the current. Here you're leaving the current, but measuring the voltage. People often say to me, well, it's just opposite. It's not. Again, in these recordings, I'm looking at inward flux of ions, and the same thing is occurring here. When I give a stimulus, influx of positive ions, you see a positive deflection, in this case from -65 to -64. This is a single synapse. the depolarization is very small, but again, it is measurable, and it tells us about the function of the channel receptor complexes that are underlying the response. So with this switch from voltage clamp to current clamp, in the same recording, the same synapse, what I did was apply a trained stimuli in this case, which mimics a nociceptor activity from, say, C fibers or A delta fibers in the periphery, the fibers that respond to noxious or painful stimuli. And as you can see, the depolarization here, and with the second stimulus, it's a bit higher. And ultimately at the fifth, it's much higher than at the first. And this is what we call temporal summation. And so this in itself was quite exciting, but my interest was to resolve how the different gluon2 subunits are mediating this temporal summation. My next experiment right off was to test the DAPV, the potent NMDA receptor antagonist. This, regardless of subtype, this will block them. And so we know immediately that this form of temporal summation, this depolarization that we see, is completely blocked by DAPV. And the AMPA receptor blocker, shown here, completely wiped out the response. So we know that this temporal summation, this micro EPSP or excitatory postsynaptic potential burst, is mediated by glutamatergic receptors, AMPA and NMDA receptors, and this temporal summation is mediated predominantly by the NMDA receptor. The question remains, though, what is the role of the subtypes in this temporal summation at this principal sensory synapse? And of course, the voltage clamp experiments indicated that there's a diversity in the gluon2 subunit. So with that in mind, in the context of diversity, in current clamp, I also have the ability to record from specific neuron types. Of course, this is not done intentionally. It's carried out in a blind manner, but I determine what neuron I'm recording from by giving current injections. And so some of the neurons I recorded were as single non-spiking, delayed phasic, tonic spiking. I would say in the last 15 to 20 years, a lot of focus on these different neuron types in the field of pain research, in which researchers achieve these recordings, these neuron recordings, characterize, and at the same time label the neuron with the attempt to try to correlate spiking activity with the morphology of the lamina 1 neuron type. In my case, I had the micro EPSP recordings as well, a lot of information in hand. And so when I was able, in each recording, of course, I had knowledge of what neuron type I was recording from. And so, for example, you can see here from the first micro EPSP in the train, it is shown here, the fast decay response single here has a very rapid rise and then a fairly rapid decay back to a resting membrane potential. And by the fifth micro EPSP in the train, again, it's still fairly low, rapid rise, fairly rapid decay from this neuron type. And in micro EPSPs recorded from delayed phasic spiking neurons, you can see the fast rise, a bit of a slower decay, and with the fifth micro EPSC, quite a bit of summation, and still quite slow, but more than the single spiking micro EPSP. And of course, with the tonic, you can see an incredibly slow decay. And by the time we get to the fifth micro EPSC, there's a considerable degree of summation. And you can see that there appears to be some form of correlation with neuron type. So at this point, the question is, what is underlying? Is it the gluon2 subtype in each case that is governing the degree of decay and summation, and does this correlate directly with neuron type? Essentially, is it neuron specific? So to interrogate the kinetics more thoroughly, what I did was leverage, with the help of Dr. Olivia Garcia, we carried out hierarchical clustering on the first micro-EPSC out of our original data set, as well as the fifth, independent of neuron type. And what we looked at was the half-width area under the curve and the decay in each case. And so just based on kinetics, not neuron type, individual of neuron type, we saw 3 groups, which is great. So far consistent with the 3 micro-EPSP data sets that we found. And what I did then was plot, based on Olivia's hierarchical clustering data, plot the 3 groups, fast, intermediate, and slow micro-EPSP kinetics. And this is for the first, this is for the fifth, and you can see here we have slow, intermediate, and fast. For the fifth, of course, it's a bit larger because there's a bit more summation in terms of the decay values and the half-width. When I plotted the same synapses, but based on neuron type, my prediction would be that they're the same, that the kinetics correlate with neuron type. And at first glance, if you look closely, it appears at first glance that it does, but if you look closely, it doesn't, right? With the black dots here, tonic does seem to correlate quite strongly with slow kinetics, but if you look closely at the delayed phasic spiking neurons and the single, they do not quite correlate completely with the kinetic values. And this is what we show right here in these pie charts. Quite a robust correlation with slow micro-EPSP kinetics correlating with tonic, but you can see with the delayed phasic and the fast spiking neurons, it's quite different. There's quite a mix of the synapses that project onto them. And so it's not so much a question of neuron specificity. I would say it's more of a question of synapse specificity, which again tells us how complex these synapses are. Now, getting to the GLUN2 subtypes underlying this, to examine that, in another set of experiments, I looked at exclusively non or single spiking neurons and interrogated the micro-EPSP responses with the known GLUN2 selective inhibitors. And in this data set, you can see that we see a considerable effect of the depressive effect on the micro-EPSP with the GLUN2A inhibitor, no effect of the GLUN2 inhibitor, and also a fairly robust effect of the GLUN2B inhibitor. Again, consistent with the hierarchical clustering that in fast spiking, single initial spiking neurons, there's a predominance of fast kinetics that's fast and intermediate that, as demonstrated here, is mediated by GLUN2A and GLUN2B. In the delayed phasic and spiking neurons, we observed an effect of the GLUN2B inhibitor, as well as the GLUN2A inhibitor, and a very minimal effect of the GLUN2D inhibitor. Again, consistent with the hierarchical clustering, showing a predominance of fast and intermediate synapses that project onto these neurons. And of course, with the tonic spiking, as you can see quite clearly here, the absolutely slow kinetics, these were exclusively sensitive to the GLUN2D inhibitor with no effect of the other inhibitors, consistent with GLUN2D mediating the slow kinetics of micro-EPSP bursts on predominantly GLUN2 tonic spiking neurons. So to summarize in a schematic, I realize that last figure was somewhat complex. But the main, what the data together show us is that there is a complexity of this principal sensory synapse. And the complexity lies predominantly in the gluin-2 subunit, and its role in the degree of temporal summation of micro EPSPs, synapses, where 2A for gluin 2A, for example, governs a low degree of temporal summation, predominantly in single spiking neurons, but also some of the synapses that occur on delayed spiking neurons. 2B plays a role in the intermediate form of temporal summation on delayed spiking neurons, but also a degree on the single initial lamina 1 neuron type. Whereas the gluin-2D NMDA receptor subunit plays a primary role in tonic spiking neurons, but also, based on some of the hierarchical clustering data, can also have synapses that form on delayed spiking and play a role in temporal summation on these neurons as well. So the main points I would like to leave you with is that there is an enormous amount of heterogeneity, complexity at the primary afferent lamina 1 neuron synapse. And this complexity derives predominantly from what we know now from these recordings in the gluin-2, NMDA receptor gluin-2 subtype that exists at each synapse. There appears to be some correlation with lamina neuron type, but I would say based on our findings, it's predominantly synapse-specific. These gluin-2 subtypes play a key role in temporal summation, as we saw with the micro EPSP bursts. And so, again, speaking to the role that these distinct synapses with distinct gluin-2 subunits, their specific role as coding units, computational units that encode synaptic input. And of course, these synapses occur on dendrites of lamina 1 neurons. So the point I'd like to share with you here is that, of course, this can play very complex roles in how sensory input is integrated from the periphery to the spinal cord dorsal horn, in this case, lamina 1, and how it's projected to the brain, to the pain sensory regions in the brain. If there is one message I can leave you with, is that these synapses, contrary to belief, are quite complex. They work as coding units, and this coding relies primarily on the NMDA receptor and the different gluin-2 subtypes that I've shown to occur in spinal lamina 1 at the primary afferent lamina 1 synapse. I would like to, as I mentioned, I'm at Sick Kids Hospital. I work with Mike Salter, and he's a great fellow, a great friend, and a lot of exciting projects going on, not only in the brain, but I'm also carrying out some exciting work in the spinal cord dorsal horn. And I would also like to thank Dr. Livia Garcia for her massive contribution with some of the hierarchical cluster and principal component analysis data. Thank you for your attention.
Thank you very much, Dr. Peter, for that fascinating presentation. So we now have some time for questions. If anyone has a question, please feel free to unmute yourself, and you can ask directly, or you can write in the chat as you wish. Any questions?
I'm gonna ask a question. just to put you on the spot. I was wondering. I know we talked about this quite extensively a few years ago, but if there is any, what is currently known on the distribution of the subunits in terms of local distribution along the spinal cord in humans? Is there a regional enrichment for some of these? Do we have the technologies at the protein level to detect differences and to understand where these processes may be more implicated in different human diseases?
Great question. So I haven't looked at that. We haven't. A colleague of mine in Ottawa, Dr. Michael Hildebrand, has the privilege of gaining tissue from humans, and so from their local hospital. And so what he has discovered is, and to your point of where in the spinal column, thoracic, lumbar, so on. So when they get some of this tissue, they get it from patients that have had obviously passed away, but some that are naive in the sense of no chronic pain conditions and some with chronic pain conditions. And they get the tissue from wherever they get it in the spinal column. So it could be sacral, lumbar, and so on. So to date, he has identified gluin-2 subunits corresponding to 2A, 2B, and 2D, consistent with what I have shared with you here. So far, it's consistent. And regardless of level of the spinal cord, the spinal dorsal horn, it seems to be consistent, which is good for basic research, such as what I'm sharing with you in rodents, because it shows that there's a similarity. We can still use our rodent models as appropriate models to study these receptors. But that's pretty much what I know of. That's pretty much it. And again, that kind of research is quite limited because it's hard to do. These experiments from humans are hard to do. But so far, it's consistent.
I guess the shout out to the spinal surgeons and the audience to help the basic researchers to
absolutely
to collect more human tissue when possible, so that we can explore more of that.
Yeah, no, for sure. Yeah.
So I don't know if I took the notes correctly, but that given that glu-N2A containing NMDRs, that they mediate fast synaptic responses, could they be a target for pain modulation? Is their function too essential for normal sensory processing?
Oh, that's a great question. So in terms of clinical, there's very little, primarily because a lot of these drugs have to be given orally or systemically. So that and of course, given the role of these receptors in development, that can pose some problems. In terms of what's known about the selective gluin-2 subtypes in pain processing in behavioral models, there has been some focus on gluin-2B, very little on 2A and 2D. At this point. And I think there is a certain interest right now because just looking at the research, there a lot more is being done in behavioral models to determine the role of these different subtypes. But I would say the field is still a bit new, a bit young on this to say specifically whether 2A, for example, plays a key role in chronic pain diseases. And I think it's quite complex given the selectivity of these synapses on distinct lamina 1 neuron types. I don't think we've resolved what that means. In fact, to that point, I don't even think we know clearly the tonic spiking, the delayed phasic, the single spiking, what role they play in processing. So it's still quite complex. It's still quite complex. Important question, though. Thank you.
Yeah. So if I wanted to develop an animal model and figure out all these wonderful conclusions that you presented here, so I think in other words, do you plan to check this in an animal model?
Of pain? Absolutely. And that is a very important question. A highly complex question in that, in fact, some of the work I have multiple papers in preparation. Some of the journals asked to carry out this research in a chronic pain animal model. And the thing is, this study that I shared with you today is approximately 8 years of research. They're incredibly difficult and challenging experiments. And so to carry this out in a model, for example, let me show you some supplementary data. So this is the data I shared with you today, the cluster data that Livia and I worked on. And this is approximately 300 recordings. Basically, one objective would be to carry out the same experiment in an animal model, but I'm not sure what it would tell us, because it may be in a chronic pain model, we have more of 2D versus 2B, or more of one or the other. That would probably be several years of research required to study this. It's massive. And speaking with Livia, that would probably, upwards of 1,000 recordings. So it's possible, to your question, to your point, to do it, but it's just a massive investment. What I am doing is to get around this is I'm in my voltage clamp recordings carrying out a pairing protocol. And for those of you who follow the long-term potentiation research in the hippocampus, this is a technique used by Roger Nicole, Robert Malenka, where they look at the EPSC, micro EPSC. Well, they look at population. I'm looking at micro EPSC. They give a pairing protocol by increasing the frequency of stimulation and depolarizing. These are phenomena that occur in chronic pain models. And then look at the magnification of the micro EPSCs following. And as you can see here in some of the experiments I've done, in fact, this is a paper in preparation, you can see quite a robust response. And in this voltage clamp experiment, I have the privilege, as I showed you earlier, I can also clamp it positive and get a very clear readout of the NMDA receptor component. And that's exactly what I've been doing. And I have a paper in preparation. Why this is important and exciting is that it's at a single synapse resolution. Okay, so I can amplify synapse and compare the NMDA component before and after. And other models, you simply can't do it. You cannot do that in vivo. That would be impossible. And if you induce a pain model in a rodent and then take out its slice and examine it, you don't know what that synapse, how it behaved, if you will, before the induction of the chronic pain stimulus, the model. So this is one approach I'm taking right now, and it's proving to be quite exciting. So very important question. I gave you a lengthy answer. I apologize, but this is how we're moving forward on this right now.
Yeah, this is clearly a lot of work, a lot of really quality work, really wonderful. Any other questions?
Yes, we miss here. Very nice work, actually. I really like it. Really appreciate the detail.
Thank you so much.
Into classifying the different types of neuronal responses. So the question was actually kind of related to your chronic pain, I guess, when you start doing the modeling, but more like on the actual NMDA receptor. So is it possible that other forms of modification can affect the, let's say, the kinetics, and not just, let's say, the subtypes? Like, for instance, GPCRs such as the mGluR, there are other GPCRs in the postsynaptic neurons. When activated, they can phosphorylate, for instance, and NMDA receptors and affect their function. So is that another layer of complexity that could be added to trying to understand how they operate in chronic pain?
Absolutely. So in our department here, also in Mike's lab, one of the main focuses historically as well has been looking at phosphorylation of the NMDA receptor. So that's been done mostly in the hippocampus, but a little bit has been shown in the spinal cord dorsal horn. And we've also looked briefly at not only SARC, but FIN, other phosphorylation molecules. And so what's intriguing about that, it could be that there's a differential role of these in the in the gluN2 subtype. That's something we've not quite fully resolved, but some of the research, we haven't published it yet, but it seems to be leaning that way. So to your question, it's not just so much the complexity of the subtype, but the kind of the phosphorylation that these subunits receive and what is phosphorylating them.
Mm-hmm.
So yeah, no, absolutely important.
Yeah.
Absolutely
Mm-hmm.
great question. Thank you. Yeah.
Yeah. So as you were talking during your presentation, I was had an internal dialogue where I was thinking, considering the heterogeneity of your gluN2 subtype configuration, do you see a future where you have subunit selection and NMDA receptor modulators that where they could be, some sort of effective strategy for pain management than what is used where you use a global NMDA antagonist like ketamine?
Oh,
Because
yeah.
our lab is more into therapy. We like find mechanism and then we run to find therapy kind of thing. So
Right, exactly. So in terms of the subunits, I don't think it's clear quite yet, not only in humans, but preclinical models, how these selective subtypes, what chronic pains they may underlie. Again, what I'm sharing with you is naive. No one has looked at this complexity in a pain model. So we don't even know. So for example, pain models, there's spared nerve injury, there's inflammation, various models. It could be that one is selective, plays a role in one, not the other. We have no clue. We have no clue. So I think it's maybe a bit, I don't want to speculate too much on that. But you do raise an interesting point about a pan inhibitor, for example. DAPV has often been considered one, but again, you cannot give that systemically. You cannot get because NMDA receptors play such a critical role, not only in the spinal cord, but in the brain cognitive functions. So giving anything systemically could be potentially dangerous. Now, you do mention ketamine, and that's receiving a lot of attention right now in terms of depression. A lot of groups are doing studies looking at the role of ketamine in preclinical models of depression. It's also used in humans to treat depression. So, and now there's a lot of preclinical research looking at ketamine in chronic pain models. So I would venture to guess, to your point of NMDA receptor inhibitor blockers, we're going to see something on ketamine potentially used for in human studies, chronic pain. In terms of the selective inhibitors, I don't think we're there yet. I don't think we're there yet. And some of the preclinical models, animal models, we're getting close to a clear understanding. 2B, for example, but as I said, 2D, 2A, it's people are doing the research on it now in behavior, from what I can observe. It's still a bit complex to speculate, though.
Slightly off the topic, but what does that have to do with memory? I'm sure like there's implications to memory, I guess.
Oh, absolutely. So, well, this slide, I don't know if you can still see it. This pairing protocol form of LTP, not at minimal stimulation intensity, but in terms of population responses. And what I mean by population, looking at many synapses. This is just an example of one. Roger Nicole, a prominent NMDA receptor physiologist, Robert Malenka, have shown ample evidence of NMDA receptors in long-term potentiation in the hippocampus, which is a synaptic, believed to be a synaptic proxy of learning and memory. Some of my early work in vitro studies looking at hippocampus, I looked at Sark phosphorylation of NMDA receptors in the hippocampus, looking at theta burst induced LTP. The NMDA receptor is highly, highly, highly critical in cognitive functions in the hippocampus, in the prefrontal cortex. Also with Sark, we showed that. So again, one has to be careful giving a potent drug affecting NMDA receptors because it can have major consequences in cognitive function. Yeah. I think one approach would be to give it locally by intrathecal injection, but my understanding is that that's somewhat limited. But absolutely. That's if we could find a way to get these inhibitors selectively to the spinal cord dorsal horn without consequences elsewhere damaging the local tissue, that I think that would hold promise. That would be quite exciting.
You have a lot actually to look into, so it's pretty interesting, very cool. Not just post-translational modification, but you have like even up and down regulation of the genes themselves in chronic pain, or
right, exactly. Yeah,
very fascinating, and thank you very much, Dr. Peter, for that really wonderful presentation and really exciting knowledge about the pain and the whole really pain system.
Thank you so much. It was a pleasure sharing this with you, and I appreciate the important questions. It was quite enjoyable. Thank you so much. Thank you.
Thank you everyone for taking their time to come to the seminar.
Excellent. Thank you.
See you next week, and thank you, everyone.
Thank you so much. All the best. Thank you.
Thank
All
you.
the best, you guys. Take
Bye.
care. Bye-bye.